Thank you very much for the invitation, and you all welcome to my talk. My talk is on artificial intelligence, machine learning, and big data for drug discovery. My name is KOJO KWOFIE. I'm a faculty at the Department of Biomedical Engineering and also hold a position at the West African Center for Cell Biology of Infectious Pathogen as a bioinformatician at the University of Ghana in Ghana. Discovery of new drugs is very, very expensive, challenging, time consuming. And at the same time, the number of approved drugs keep declining steadily. And it's been shown that toxicity accounts for 90% of withdrawals. And major early stage termination usually relate to suboptimal efficacy and safety. Sir James Black, a Nobel Prize winning pharmacologist, advocated drug repurposing, and he made a famous statement, and the statement is the most fruitful basis for the discovery of a new drug is to start with an old drug. On the average, it takes about 10 to 15 years and roughly around $1.5 billion from initially presenting the abstract concept, the proof of concept, to putting the drug into market. It's very, very important, around 1.5 billion. And then also almost 10% of repurposed drugs so far have won regulatory approval, and then 70% of them are in clinical development. What is the role of artificial intelligence, AI, in drug discovery? Artificial intelligence and its application start right from the identification of potential lead compounds, moving it forward to clinical trials, and even at the approval stage when it comes to the selection of population for clinical trials, understanding the pharmacology of the drug, and also understanding the molecular mechanisms and then the binding mechanisms involving the drugs. Artificial intelligence has been widely used in drug research. And what is artificial intelligence? It's basically any technique that enables computers to mimic the human brain, and it encapsulates machine learning, which is simply a data analysis technique that automates analytical model building using algorithms that can learn iteratively from data. So these computer algorithms can identify hidden insights without being explicitly programmed. And what are some of the categorization of machine learning? You have supervised learning, you have unsupervised learning, you have reinforced learning, and there's another paradigm which is widely used nowadays, that is deep learning. So what are some of the various techniques used in machine learning for drug discovery? You have classification, you have regression, you have clustering, feature finding, decision making, and execution. And it's very, very important. These are applied when it comes to the prediction of the pharmacology of the drugs, efficacy of the drug, the disease subtype, discovery, also for the identification of disease targets, drug target, and also for the design of compounds using the de novo technique. That's basically de novo design of compounds. So once you have done the de novo design of compounds using these algorithms, you can then synthesize these compounds and test experimentally. It's very, very important, and this makes drug discovery processes very easy, very efficient, very fast, less cumbersome, less expensive. Because instead of buying thousands of compounds and testing these compounds experimentally, in vivo and in vitro, which is very, very expensive, you can use these computer models, algorithms, application tools to screen these thousands or billions of compounds, shortlist or prioritize potential lead compounds, few of them, which you can do a lot of pharmacological predictions on them, try to understand their mechanism of binding, their mechanism of their mechanism of action, their biological activity, and you can do all this in silico in order to end up with few compounds that you can purchase and test, thereby cutting down the cost and time required for drug discovery. What are the input for these machine learning models? The inputs are basically the cheminformatics features of the compounds, as well as the bioactivity data sets that you obtain from databases such as PubChem, Chembo, and then binding DB, and then you use this to train these algorithms, and these algorithms will learn these features in order to be able to predict the bioactivity data sets of new compounds that you provide the algorithm. So in this instance, the model once built can be used to screen and prioritize compounds. My group published a review article on the prediction of anti-schistosomal small molecules using machine learning in the era of big data. The review article basically looked at the application of machine learning in drug discovery, and one of the important paradigm, is the source of data, and the paradigm is big data. And what are the sources of data for training the algorithm? You can obtain data sets from PubChem, Binding DB, Chembo, SchistoDB, Drug Bank, and all this you can obtain data or features about compounds. And then you have the protein data bank, which provides the three-dimensional structures of protein which have been experimentally solved, and you can extract features from these structures. And then these features will be used to train these algorithms, basically in order for the algorithms or models to be able to predict the bioactivity of new compounds. Another review article published by the group looks at Outwitting an Old Neglected Nemesis, a review on leveraging integrated data-driven approaches to aid in unraveling of leishmaniasis of therapeutic potential. So how do you use integrated data in order to build machine learning models to aid you in identifying potential drugs or therapeutic molecules? So one can obtain features from drug targets, from cheminformatics databases. One can also compute pharmacophore models for training. You can also experimentally elucidate the bioactivity of compounds, and all this could be used to build the machine learning models. Recently, we published a paper on the development of deep neural networks to predict inhibitors of schistosoma mansoni. We also developed a proteochemometric-based support vector machine model for predicting anti-tubulin compounds. The support vector model has been implemented as a web-based application known as the TubePred, and this offers the opportunity for scientists to be able to run or test their compounds or screen their compounds online. But the algorithm is also available for users to screen compounds on their high-performance computing systems or their local computers. So what are the input files? The input files are basically the SMILES format of the structures of the compounds and the UniProt ID of the proteins, and you click execute or compute, and it will predict the bioactivity of the compounds with confidence score. There is also a user tutorial manual available. and also frequently asked questions. For example, what is TUPRED? What is proteochemometric methods? What are the input data sets? And then the interpretation of the confidence score. The group also developed Ebola-Pred, a machine learning-based web application for predicting cell entry inhibitors of the Ebola virus. And this is very, very popular. The table shows the performance of the various models based on the average accuracy, precision, recall, F1 scores, and balanced accuracy values after cross-validation and prediction on internal test data. This slide shows the ROC curve for the random forest model with an area under the curve value of 0.95, which is reasonably valid. Celeron, which has been experimentally determined as inhibitor of Ebola virus, was predicted using random forest to be a bioactive compound with a confidence score of 0.75, and torimifene, which is also known to be an inhibitor of Ebola virus, was also predicted as active with a confidence score of 0.89, also within the applicability domain. So this is basically the user query interface of Ebola-Pred. So you can upload your compound structure in the SMILES format, and you can compute the bioactivity using random forest. The results are ready. It provides the bioactivity of the compound, whether it's active or inactive, with confidence score, and whether it's within the applicability domain or not. You can also use logistic regression to compute the bioactivity. And the result is within the applicability domain. You can also use support vector machine to compute the bioactivity to determine whether the compound is a potential Ebola virus entry inhibitor. It also provides the user manual as well as the frequently asked questions, which explain concepts such as a confidence score, the applicability domain, and how datasets can be downloaded. This is a paper where we use computational approaches to predict potential novel anti-Ebola virus protein VP35 natural compounds as potential inhibitors. So this slide basically demonstrates virtual screening using molecular docking. So this is a typical protein structure which is a cartoon representation of the protein structure, and this is a surface representation of the protein structure showing potential binding pockets or the active sites. And this is a typical ligand ready to dock within the active site or the binding pocket. So that is the docked compound. And virtual screening can be done using lot of compounds. So you find the compound or the small molecule docked within the active site pocket. And this a wireframe representation showing the compound docked in addition to intermolecular bonds. So you find hydrogen bonds formed with critical amino acid residues required for binding within the active site pocket. We also use computational techniques to identify putative furin protease inhibitors, which have the potential to be developed as therapeutics against COVID-19. We also use computational techniques to identify potential anti-SARS-CoV-2 natural compounds of African origin as potential antiviral compounds. We also targeted the COVID-19 induced cytokine storm to identify potential anti-inflammatory natural products targeting the P38 mitogen activated protein kinase with the hope of identifying potential COVID-19 therapeutics. So this diagram is basically a graphical representation of the step-by-step approaches used in identifying probable lead compounds for experimental testing. So first, you generate a library from natural product databases. You pre-filter using physicochemical profiling such as the Limpinski rule of 5 to identify drug-like compounds. You dock these compounds or pre-filtered compounds against the drug target, and then you further shortlist using the binding energy which corresponds to the binding affinity. Then you do binding interaction analysis, you do biological activity prediction, and then biological mechanism prediction, molecular dynamics simulation, and then you test in silico computationally the physicochemical profile and then the toxicity profiling in order to arrive at probable lead compounds for experimental testing. So this diagram is basically a methodology schema we developed for one of our work, where we generated a library of compounds for screening. We obtained the compound from FDA-approved drugs, natural product databases, and then a list of previously pre-filtered compounds obtained from machine learning study. And this integrated library was used to screen drug targets using molecular docking, and compounds were pre-filtered or further shortlisted using binding energy, binding affinity. The mechanism of binding was characterized looking at the interaction between the ligands and critical amino acid residues, interactions such as hydrogen bonding, hydrophobic interactions. We also look at predicting biological activity, predicting molecular mechanisms, and also you do molecular dynamics simulation to understand parameters such as the root mean square deviation of the receptor ligand complex. So this is a plot of the molecular dynamics simulation showing the root mean square deviation and the time in nanoseconds. We also undertake molecular dynamics snapshots, sometimes at 25 nanoseconds interval of the various complexes to understand the binding modes, the stability of the complexes, the flexibility, the rigidity. You can also compute that for root mean square deviation, radius of gyration, and also root mean square fluctuation. So this shows you typical a snapshot at 0 nanoseconds, 25 nanoseconds, 50 nanoseconds, 75 nanoseconds. And these parameters enable you to identify plausible lead compounds. This table basically provides the names and 2D structures of some of the compounds that we have identified in our study. So as part of our research, we combine artificial intelligence, machine learning, big data. We develop algorithms. So we have been successful in developing novel algorithms, models. We have used this to screen compounds. We have used existing techniques, optimized existing techniques like molecular docking, virtual screening, bioactivity prediction, and we've used this to identify potential lead compounds, which we've tested some. Some have been successful, which we are optimizing, and then we hope to develop this further beyond in vitro testing, in vivo testing, and hopefully we can have compounds for clinical trials. So thank you very much for listening to me. We have been doing a lot of our computational work using Zuputo, which is a high performance computing system, a Dell EMC system. It has 6 nodes, 12 GPUs, and then 216 CPUs. I'm grateful to my collaborators, Professor Michael Wilson, who is at the Noguchi Memorial Institute for Medical Research, University of Ghana, and then Dr. Welton Miller, who is at the Loyola University Medical Center, and Prakasha Kampai, who is at the Infectious Disease Mayo Clinic in Florida. And I'm also grateful to my research assistants, graduate students, and other collaborators. Thank you very much for listening.